Good morning, class. I hope you had an excellent Thanksgiving break. We are picking up with what we started right before Thanksgiving break, which is Unit 4, Heredity. Pink and purple classes, you had the chance to join with your class in discussion of the article that everyone had to read on nature versus nurture and what actually shapes who we are. Is it our genes or our environment? And what we came to see at the end of that discussion was it's some of both, that there are genetic factors and there are environmental factors that impact ultimately who we are. The essential questions of this unit number 4 are: how can parental traits predict the likelihood of characteristics of our offsprings? And what tools can we use, including Punnett squares, pedigrees, and karyotypes, to analyze genetic inheritance patterns? So we're going to be looking at some specific phenotypes and traits in this unit as we wrap up quarter 2. Okay, so a quick caveat. I asked you to read an article on Monday called Oversimplifying Genetics. And the reason I wanted you to read that is because sometimes the way that we teach an introductory biology class actually promotes misconceptions about how genetics really works, because it's not simple. It's really, really complex. And so I want you to be clear that there typically isn't one gene that affects one physical characteristic or trait. Often it's many genes and how they interact with environmental factors, switching genes on and off, that ultimately shape our phenotypes, or the characteristics that make us who we are. If you haven't read that article yet, please make sure you do so and read it closely. If you don't understand what it's talking about, I encourage you to ask me questions because I want you to leave this class with a true understanding of genetics. And I fear that some of the assignments that we do will promote this idea that one gene controls one trait. And that's just really not the case in most situations. There's many, many phenotypes. For example, there's tons of different versions of eye color in our population. And that's because it's not controlled by a single gene, it's controlled by many different genes and the way they interact with the environment. So keep that in mind as we move through this unit. There, just like our other units, is a lot of new vocabulary. So make sure you have your notebook out and you're taking notes on this lecture. I'm going to do a quick review of some vocab before I dive into the content for today. So heredity is the transmission of traits from parents to offspring. Sometimes we use it interchangeably with the word genetics, but that's going to be the study of those traits. A few words that you already know: a chromosome is made up of both DNA and protein, and it has many different genes on it. So a chromosome is made up of thousands of genes, and a gene is a specific sequence of DNA that controls the production of a single protein. And this is what we learned last unit when we were learning about the decoding of DNA. An allele is a version of a gene, which is represented by letters. So because there are different traits, if we're talking about, say, the gene for the height of a plant, the gene could be for plant height, but then the version or the allele of that gene would be a taller allele or a shorter allele. And that would be based on a few variations in the genetic code, or the order of the nucleotide bases, as you learned last unit. Genotype. Okay, these are recaps. The genetic makeup of an organism. Each genotype is represented by 2 letters or alleles. And so it could be a capital A, lowercase a, 2 little a's. And that's because you get one of those alleles from each of your parents. A phenotype is what an organism actually looks like. It's the observable or physical expression of the genotype. So the genotype determines the phenotype, and you could have a tall, short, green, brown hair. All of these characteristics are examples of phenotypes. An organism is considered homozygous if it has the same allele for a given trait. So that means it got two dominant alleles, one from each parent, or it got two recessive alleles. So if we're representing a specific gene, it could be two capital letters to represent dominant, two lowercase to represent a homozygous recessive. But it just means you got the same allele from each parent. Heterozygous means you got two different alleles from a parent. And that's when we have to investigate which allele is going to be stronger or be more dominant. A heterozygous genotype would mean a capital letter and a lowercase letter. So it means that the version of the trait that you got from the mom and the dad are not the same. A dominant allele is a form of a gene that will mask the expression of the recessive gene. It is always represented by a capital letter. A recessive allele is a form of the gene that is not expressed when in the presence of a dominant allele. And so to have a recessive trait show up in the phenotype, you need to have both alleles be recessive or homozygous recessive. That means you got both recessive alleles from the mother and the father. And this, if you think back to the last unit when we were exploring albinism, is why the mother and the father of the children that had the trait of albinism had normal skin color because they were carriers of a recessive allele, but it was being covered up by their dominant allele that produced melanin. Now, the offspring that ended up with the albinism phenotype would have received that recessive allele from both the mother and the father, always represented by a lowercase letter. And this is what I was just saying. To be expressed in the phenotype, the recessive trait has to be inherited from both parents. Okay, and the last vocab word is gametes. These are sex cells, so we call them egg, sperm, pollen, ovum if we're talking about plants. Sex cells have half the number of chromosomes as regular body or somatic cells, and that's because they're going to join during fertilization with a different sex cell to form an offspring zygote. Okay, so the complex version is that both genetic and environmental factors would affect, say, the height that a plant would grow, right? It needs certain environmental factors like sun, light, enough space, but there's also going to be genetic predisposition towards being a taller plant or being a shorter plant. And so today, to, again, simplify the genetics so we can understand it, we're going to be focusing on just the genetic side of what is affecting the phenotype, the height that a plant would grow, okay? So just to start, leading from last unit, sexual reproduction is one of the main reasons that we have so much variation among our offspring. Towards the end of last unit, we learned of a process called meiosis. This is how sex cells or gametes form. And it goes through 2 rounds of cell division, which allows for those sex cells to have half the number of chromosomes as the parent cell. So in a human being, we have 46 chromosomes in our normal cells, or 23 pairs, and a sex cell, our egg or our sperm, have only 23 chromosomes. Now, each daughter cell, by the end of meiosis, is different from the others, and that allows for a wide variety of offspring because of the variation that meiosis creates in the sex cells. There's also an event that occurs called crossing over that adds even more diversity in these traits existing in sex cells, and you should have learned that in the last unit. Okay, so we can think of the parent chromosomes as having both parents have the same genes on them at a specific chromosome. And let's say this male chromosome has a recessive allele for plant height, and this female parent has a dominant allele. So the parent has 2 sides of a chromosome, but when a sex cell is created, it's only going to pass down 1/2 of that chromosome. And so depending on which half you get, you're going to have a variety of offspring in the children or offspring of that genetic cross between half the mother and half the father's traits. Okay, so another way to try to re-say what I was just saying. I know this is complicated. So let's say that this first cell here represents the two sides of a single chromosome, say from the father plant. So this father plant is heterozygous. It has a dominant allele and it has a recessive allele. That parent is going to go through the process of meiosis when it makes its pollen, which is the plant version of sperm. And that process of meiosis leads to the formation of these pollen. It can either have the dominant allele, because that's what it had in its original cell, or it could have the recessive allele. This is what is going to get passed down to the possible offspring. This process is called Mendel's law of segregation, but it refers to meiosis, and we use it when we make out Punnett squares. Now, a Punnett square is what we use to predict the possible phenotypes of offspring based on the parent genotypes. It is a prediction. It is not a guarantee of what the offspring will be, but we are going to take the parent genotypes, so those capital or lowercase letters, determine what type of gametes will form by the process of meiosis, as we saw in the last slide, and then cross those with the other parent to predict this is the possibility of having a tall offspring or a short offspring. Okay, so let's look at this example because I have to keep this to 15 minutes or I get cut off. Parent number 1 is homozygous dominant. During the process of meiosis, when the sperm or pollen cell is made, it can either make a capital T, and then it can also make another capital T, because when these separate, that's all that parent carries. So we write one parent's meiosis on the left side, and then the other parent was a homozygous recessive parent. And again, when it goes through meiosis, it can form lowercase t, lowercase t. Now these boxes represent the possible offspring. So I'm going to call these fertilization events. Depending on which egg and sperm gets inherited, these are the new offspring that could be made. And because the parent 1 was homozygous dominant and parent 2 was homozygous recessive, when they cross, you can only end up with completely heterozygous. It has to get that capital T from parent 1 and that lowercase t from parent 2. And so 100% of the genotypes of offspring are going to be heterozygous, or capital T, lowercase t, and 100% of the phenotypes are going to be tall, because even though that recessive short trait is carried, it is going to be covered up by the tall trait. Now let's say these offspring reproduced with each other, and now we have two heterozygous parents, which means they have both versions of the alleles in their parent cells. When they go through the process of meiosis to create their sex cells, they can either create a capital T gamete or a lowercase t gamete, and the same for parent 2. And that means when we do the cross, we have a possibility of getting capital T, or homozygous dominant, lowercase t, capital T, lowercase t, or heterozygous, or lowercase t, lowercase t. And therefore, the genotype is going to be 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. Sometimes that is written as 1, 2 to 1. And the phenotype, which has to do with the traits, means that 75%, both this one, the capital T, and lowercase t, capital T, are going to be tall plants. 25% of the offspring would be short. So sometimes we represent that with 3 to 1. Okay, I'm running out of time here, so this is my last slide. What I want to emphasize is that a Punnett square is a prediction of a probability. Each time mating occurs, the same probability or possibility of 75% tall, 25% short exists each time. So say you flip a coin, 50% of the time, you're going to get heads 50%, tails 50%. If you flip it again, just because you just flipped heads, you still have the same 50% chance of getting heads again, okay? So the same thing works in our Punnett squares. The probability remains each new time the parents mate. So you might end up with 100% tall plants, even though there's a probability that 25% of them could be short.